with the next project, we go from biochemistry to biology. The second presentation is by James Howe from Regina High School in Iowa City.
Hello, my name is James Howe. I'm from Regina High School in Iowa City, as he just said, and thank you for the opportunity to present my research to you today. The title of my project is BMP-RNA Mutations in Juvenile Polyposis Affect Cellular Localization. Colorectal cancer is a major medical issue today. It is the fourth most common form of cancer in the U.S. and the second deadliest. It has a 5-year survival rate of about 65%, and the chance of any individual developing colorectal cancer in their lifetime is about 5%. 5 to 10% of all colorectal cancer cases are inherited. It is from these inherited disorders that most of our understanding of colorectal cancer's genetic basis comes from. One such inherited disorder is juvenile polyposis, which we will be discussing today. Juvenile polyposis syndrome, commonly abbreviated as JP, affects approximately 1 in every 100,000 people. It is a genetic disorder inherited in an autosomal dominant fashion. This is an example of a juvenile polyposis kindred, the largest known kindred, which actually originates from Iowa. On this diagram, everyone shown in black developed polyps or cancer, and all in gray are at risk. In this kindred, 18 people developed cancer, 12 in the colon and 6 in the upper GI tract. People who have inherited the disorder are estimated to have a 39% lifetime risk of developing cancer of the gastrointestinal tract. Juvenile polyposis is defined by the presence of multiple hamartomatous polyps in the gastrointestinal tract. One such case is up on screen right now. This is an image of a colon removed from a patient with juvenile polyposis. Note the pinkish red polypoid masses here, which are the juvenile polyps upon the colon's lighter healthy mucosa represented here. If we look at these polyps under a microscope, we can see several peculiar features. There's a normal mucosal layer on top where colon cancers usually develop, but below there's an expanded lamina propria infiltrated with lymphocytes, cystically dilated glands, and dense stroma. Interestingly, such large phenotypic changes are brought about by simple mutations in one of two predisposing genes. The first is SMAD4, located on chromosome 18q21, and is commonly lost in cases of pancreatic cancer. The second is BMPR1A, located on chromosome 10q23, which is a cell surface receptor. Both of these genes account for approximately 25% of recorded JP cases. The genetic origins of the other 50% of juvenile polyposis cases are currently being investigated. BMPR1A, full name bone morphogenetic protein receptor 1A, is a transmembrane serine threonine kinase receptor in the TGF-beta superfamily of proteins. Pictured here is the tertiary structure of BMPR1A. This depicts the BMP signaling pathway, which influences many cellular processes, including cell growth, differentiation, and apoptosis. BMPR1A plays a very important role in the pathway. It forms a complex and is phosphorylated by BMPR2, and it forms a complex with a BMP, specifically BMP2, 4, or 7, which acts as a ligand for the receptor complex. This complex then proceeds to phosphorylate SMAD1, 5, or 8, and this SMAD then binds to SMAD4, and the complex enters the nucleus. Here, this complex acts as a transcription factor in conjunction with DNA-binding proteins. In previous studies of 102 separate JP patients in our lab, 22 distinct BMPR1A mutations have been found. Of these, 7 were small deletions leading to frame shifts, 6 were nonsense mutations, 1 was a splice site mutation, and 8 were missense mutations. The mechanism by which frame shifts and nonsense mutations bring about disease is relatively clear. The protein will be truncated, which leads to aberrant function or premature degradation. More challenging, however, is finding the mechanisms by which missense mutations cause disease. This could occur by changing a critical amino acid, which could potentially lead to changes in the protein's tertiary structure, which could alter its interaction with other proteins or perhaps even target it for degradation. The focus of this study was on the 8 missense mutations highlighted in blue, which occurred in 3 separate and distinct protein domains shown in red, and how they might cause juvenile polyposis. The objective of this study was to study the impact of missense mutations on the cellular localization of the BMPR1A protein and its impact on the phenotype of juvenile polyposis. To study these mutations in an in vitro system, a BMPR1A expression vector was used. The expression vector consisted of a BMPR1A gene with a CMV promoter upstream and green fluorescent protein commonly abbreviated as GFP downstream. The BMP-RNA created from the vector was expressed with the GFP tag at its C terminus. This led for visualization of the protein's cellular location under confocal microscopy. First, site-directed mutagenesis was utilized in order to recreate specific JP patient mutations in the vector. These vectors were then verified for accuracy and efficacy by direct sequencing, and then the vectors were transfected into a human embryonic kidney cell line known as HEK293T in the presence of BMP4, which helped to stimulate the BMP pathway in the cells being viewed. After transfection, the cells were incubated for 48 hours. Before being viewed, the slips were fixed in 10% formalin to anchor the cell to the substrate. Viewing and image capture performed by confocal microscopy, and the images were adjusted for contrast using the ImageJ program. ELISA was used to quantify the amount of BMP-RNA in the cell, and mutations were also analyzed in silico by 7 different predictive algorithms to predict how deleterious the mutations would be to protein function. A qualitative scoring system was developed to classify the images. The images were assigned a score based on the interpretations of the image by multiple blinded independent judges. Each judge would score each image on a scale from 1 to 3. A score of 1 would indicate strong membrane staining. A score of 2 showed that BMP-RNA could be found mainly in the cytoplasm, but with a small amount reaching the cell membrane. And a score of 3 would indicate that BMP-RNA predominantly localized to the cytoplasm. An image's cumulative score would be determined by using the mean of the judges' scores of the image. A judging system was used because no useful software could be found to discern between membranous and intracellular staining. All 8 missense mutations were successfully recreated via site-directed mutagenesis. This is the list of the mutations selected for the experiments with exon data, specific base change, and residue change noted. Of the 11 exons in BMP-RNA, more than half are represented, with the location slightly skewed toward the beginning of the gene. 1 mutation was in the signal peptide, 4 were within the MH1 domain, and 3 were in the intracellular kinase domain. This slide demonstrates the results of the in silico analyses with the 8 different mutations on the left and the results of 7 different predictive models on how damaging the mutations might be listed across the top. There's a lot of data on this slide, and I apologize for that, but to summarize, all mutations were found to be deleterious in a majority of models. So this is the image obtained from the transfection of the wild-type control vector at 20 times magnification. The nucleus had a blue DAPI stain applied to it, and the green on the image is GFP. The transfection efficiency was approximately 20% across the board for all images captured. This image received a score of a 1. This is the same sample, but at 63 times magnification. Note the halos formed by GFP here on the cell, and these linear arrangements of GFP indicate that the protein was lined up along the cell membrane. This image was scored as a 1 again. The cells pictured here have a mutation in the MH1 domain of BMP-RNA. This image was scored as a 2. This is the same image at 63 times magnification, and at first glance, cytoplasmic localization of BMP-RNA is the most obvious, especially in the cells up here. However, upon further inspection, membranous localization of BMP-RNA can be seen, most specifically in the bottom cells here, but it wasn't to the degree that there was of cytoplasmic localization. But because these 2 modes of localization were found in the sample, albeit not to the same degree, this image did receive a score of 2. The mutation pictured here also had a mutation within the MH1 domain of the protein as well. This image was scored as a 3. This is the same image on higher magnification. Here the difference is even more noticeable. As you can see in the cells in the center of this image here, the GFP can be seen in patches all over the cell, showing no clear pattern of localization to any specific feature. All that can really be said about the pattern of localization in the protein is that it seems to be intracellular. Again, this image was scored as a 3. This table summarizes the localization scores of each sample. Every single mutation received a score at or above 2, while the wild type was scored as a 1. This shows clear differential localization between the mutant and wild type BMP-RNA. It also shows ELISA data of each one, reported as a percentage of the wild type. Generally, the amount of protein stayed relatively normal across all mutant samples, with one exception at 1A to C M1L. The protein actually experienced front-end truncation here, removing the ELISA antibody's binding site. Excluding this mutation, the mutant assays had a mean percentage of 101.6% of wild type. This suggests that levels of protein are not a significant element in the signaling alterations examined in the study. Multiple conclusions were able to be reached based on the results of these experiments. The first was that all mutations were deleterious by way of in silico analysis. ELISA data showed that the amount of protein was not a significant factor in the signaling alterations in this study, and BMP-RNA was not degraded prior to localization. The images also showed clear cytoplasmic localization of BMP-RNA, of the BMP-RNA mutant, which is very different from the wild type's membranous staining. As a whole, the conclusion was able to be reached that mucin mutations in the gene BMPR1A do affect the in vitro localization of its corresponding protein. These results provide a mechanism for impaired BMP pathway signaling in patients with juvenile polyposis. A review of the literature suggests a few possible mechanisms for these findings. First, a study done by Ruder and Conchana et al. Examined similar mutations in BMPR2, BMPR1A's membrane binding partner that shares a significant amount of homology with it. And these showed that mutant protein accumulated in the endoplasmic reticulum, not making it to the cell surface. Second, a similar study done on the same protein by Sobolewski et al. Showed that the treatment of the cells with chemical chaperones like thapsigargan would actually restore mutant receptor localization to the membrane. I found these studies to be intriguing and relevant to the current study, and they certainly provide fertile ground for further research. I envision performing co-studying experiments to confirm localization of mutants to the endoplasmic reticulum and possibly do chemical chaperone treatments which could have possible therapeutic implications for patients in the future. And before I end, I'd like to thank a few people and groups. I'd like to thank Jim Howe, my mentor. I'd like to thank Fadi Dadale, Scott Sherman, and Dong Hong Wang, who all assisted at various points in the project. I'd like to thank the College Board and the Siemens Foundation for putting on this competition. I'd like to thank George Washington University for hosting the competition. I'd like to thank all of you for coming out to listen to this presentation today. Thank you.